Okay, today we're going to talk about the very first chapter of my new book. And the title of this chapter is My Metamorphosis, because this is all about the stage in my career where after studying for 7 years to become a physician, I really didn't like it. And I made a decision sort of halfway through this chapter 1 where I said, it's got to be a better way. So we start out in 1970. If you can think that far back, at which time I left Yale New Haven Hospital, where I was an intern and a resident, and having graduated from medical school in 1968. So 68 was the year in terms of the 20th century, as far as I can tell. Although at the time, I don't think any of us knew that this was going to be such a monumental year. Anyway, I was really in the throes of residency training between 68 and 70 on every other night and every other weekend. And basically, I was sleep deprived throughout the whole time. I barely even knew my own name, let alone the names of all of my patients and so forth. But the good news is that, the academic year in medicine goes from July 1st to June 30th. And so towards the end of June in 1970, my wife and I loaded all of our belongings and our children into our car and drove down to Baltimore, Maryland, where I joined a branch of the National Cancer Institute, which was called the Baltimore Cancer Research Center, and abbreviated, as all things happen in medicine and science, as the BCRC. And on arrival, they had us all the new recruits gathered into a room, and they told us which service we were going to be assigned to. And so they went through the whole thing. There were 11 of us. They went through the whole thing. I think I was last. And they finally got to me and they said, "Will Smith, you're going to be on neurosurgery." And I said, "Neurosurgery?" I said, "I have no idea. I don't know anything about neurosurgery," because I was internal medicine at Yale. And they said, "We don't care. You're in the Army now. And so you're going to be a neurosurgeon because we need you. We need a warm body on neurosurgery." They didn't really say it that way, but that's what they meant. What they were doing at the BCRC at that time was that one of the full-time faculty or professors or whatever was a neurosurgeon, and he was investigating new chemotherapeutic agents in patients that had brain tumors. So I learned, what I did learn in that first year on the service is I learned how to do a really competent neurological exam. I also learned that I didn't want to have anything to do with neurosurgery or neurology or anything like that. The whole thing was really depressing to me because the patients were really sick, as you can imagine. One of the highlights of the whole time was that I left Yale, where I was on every other night and every other weekend on call, which meant I was in the hospital at that time, taking care of patients all night long. I'd come home the next day at the end of my shift, and I was just completely exhausted. I ate dinner, I went to bed, got up in the morning, and it just kept on going like that. However, at the BCRC, it was great because there were 11 of us, and we had to cover the ward that they had. And so we were only on every 11th night. I mean, I thought I was in heaven and had lived and died. It was really great. And of course, my family, my wife, and I had 2 little kids by then, Carrington and Kelvin. Everybody in my family has a K name except my wife, Lynn. So Carrington and Kelvin were 2.5 and 3.5 when we left New Haven and went to Baltimore. And during that first year, taking care of the brain tumor patients, I also took care of some children that had acute lymphocytic leukemia or lymphoblastic leukemia, and that was much more interesting to me because that was pediatrics because they were kids, but it was a medical phenomenon rather than a surgical phenomenon. And I had about halfway through that year, I had a patient, a young girl. She was about 11 or 12 years old, and she had acute leukemia. What we call ALL. And she'd had leukemia for like 4 or 5 years. And she'd been on combination chemotherapy, meaning the 4 or 5 different chemotherapeutic agents off and on throughout that 5-year time period. The drugs would get her into remission so that there was no evidence of leukemia and they could send her out of the hospital. But then inevitably at that time, she relapsed and had to come back in and undergo very high doses of chemotherapeutic agents And one of them was a natural hormone of the body that was called glucocorticoids or corticosteroids. And it was known at the time, the steroid hormones were isolated in the 1950s. And so this is 20 years later in the 70s. And it was known that these glucocorticoids or corticosteroids were immunosuppressive. And they were also effective agents against lymphomas and leukemias, lymphocytic leukemias, and causing the lysis of these cells in the body if you administered very high doses of these things. But these steroids were really toxic. They're much more subtly toxic than most of the chemotherapeutic agents, which caused you, after you got your dose, and usually acute nausea and vomiting, and then your hair would fall out and all kinds of bad things would happen to you. The steroids were a little bit more subtle than that. They altered the metabolism of all the major nutrients that we put into the body, meaning proteins, fats, and carbohydrates, sugars, and starches. And with the proteins, they basically slowed down or stopped the use of these proteins for building up muscles and other structures in the body. And the body started tearing those things down. And so what happened to you is that you had muscle wasting. And at the same time, they stopped the metabolism of fats as a nutrient that you could burn for energy. And so you developed a characteristic, what they called a Cushingoid syndrome, with a moon faces and a great big deposition of fat in the stomach, deposition of fat in the back called a buffalo hump. All those things happen. And in addition, you didn't metabolize sugar and starch normally either. So you developed diabetes, essentially. And so this patient of mine was miserable. Her family was miserable. Her mother, they'd been going through this for 4 or 5 years. They had 2 or 3 other kids at home. They were traveling long distances to go see her in the hospital. So she was left alone a lot at the time and so forth. To make a long story very short, she ultimately died. And she died of an infection or infections because of the fact that the steroids were immunosuppressive, as well as the chemotherapy was immunosuppressive, as well as the leukemia was immunosuppressive. So this poor kid didn't have a chance. And I said to myself, I was really upset when all that. My wife remembers that one of the other clinical associates, which was in my peer group, that's what they called us, called her and told her, you better watch Kendall because he is very depressed about this whole thing. And I came home and I told her, I said, this is crazy. I said, the patients are really sick when they walk in the door, and then we give our toxic drugs to them and they get really sicker, and then they all die. There has to be a better way. And it was during that time then that I decided to look for a better way. I began my metamorphosis from a caterpillar into a butterfly is the way I think about it. So I started doing a lot of research and reading and so forth in cancer and leukemia. And I realized as time went on and as I gained more and more information that nobody really had a clue what cancer was, what a cancer cell was, and how that cancer cell, they could identify it under the microscope, but they didn't know how it differed from a normal cell. Say it's a lymphocyte, so it's a leukemic lymphocyte or a normal lymphocyte. They looked different. They had a different morphology, but they didn't have a clue as what was going on inside the cell, what was going on with the molecules and the genes and all these different kinds of things. So this was the beginning, really, of the golden age of biomedical science, because during the 1970s, 73, gene cloning and genetic engineering was discovered. And then, and we'll get into some of those things a little bit later on. And then in 1975, monoclonal antibodies were developed and reported. And those 2 discoveries in the 1970s allowed the science to progress from the cell, looking down the microscope at the cell, and you say, okay, yeah, they're there, and I can count them. And some of them are funny looking, but that was about it. So it was really a descriptive process. Cancer was, at least at the scientific level. And so I decided that it was going to be very difficult to try to understand cancer by studying cancer cells, only cancer cells. So I figured that what we needed to do was to figure out what regulated normal cells. say, normal lymphocytes. And what regulated whether they proliferated and they grew and multiplied, or whether they didn't, and so forth and so on. So I went back to the library and I started studying normal cells. And I found out that the only real model system that the scientists had been really focused on since the mid-60s were mouse embryo fibroblasts. And one of the reasons they decided to work on these cells was that they were easy to grow in tissue culture. However, if they started looking carefully at these, what they thought were normal cells, they found that they had all kinds of chromosomal breaks and chromosomal duplications and triplicates and so forth and so on. These cells were not normal. They were very, very abnormal. Well, the whole field of people that was focused on normal cells and normal cell growth were using these cells. Well, I said, I was just young and naive and brash. I thought, well, this is crazy. This is just stupid. Why would you do that? And so along came a fellow towards the end of that first year in the clinic. I forgot to say that the deal was in the National Cancer Institute during the Vietnam War is that if you were accepted into the National Cancer Institute, they made you then a lieutenant commander in the U.S. Public Health Service. I didn't really know what that was, but I found out. And actually, the U.S. Public Health Service was started in 1798 by John Adams to take care of the merchant marines, because they were being plucked out of the oceans by all the enemies, and plus pirates and all kinds of things were going on back in those days. And they needed, so they sent these poor merchant marines out there, and then they would come back all bunged up. And so they started the U.S. Public Health Service, and that's it. So that was a good thing during wartime, because that meant instead of going to Da Nang over in Vietnam, I went to Baltimore. Little did I know, Baltimore was almost as bad as Da Nang. I moonlighted while I was there, and I saw more gunshot wounds in the emergency rooms of a little hospital in West Baltimore than I think I would have seen in Vietnam. Anyway, about towards the end of that year, there was one of my fellow clinical associates by the name of Leonard Chess, invited me to come and work in his laboratory. Wasn't his, but he was one of my peers during the next year, the second year, because the deal was you do 1 year on the wards, and then you can go into a research laboratory and do research for the second year. And that was down. Basically, that was a sweet deal. So Lenny said, I'm working on these lymphocytes with this fellow by the name of Michael Martinet over in the next building. He said, why don't you come over and I'll explain it all to you and you can check it out and so forth. And so I did that, and I joined their laboratory, and Michael Martinet was the head of the lab, and he was trained clinically as an allergist. And he was a New Yorker from Brooklyn, and he still had a trace of the Brooklyn accent. But the good thing about Michael is that although he was trained as an allergist, he did a postdoctoral fellowship at the Scripps Research Clinic in La Jolla, California. And he came to the BCRC in Baltimore in 1967. And so this was 1971. That was only 4 years. But he set up his laboratory there, and Lenny had been in the lab. I don't know how he got a deal, but he circumvented. He only had like 3 or 4 total patients in that first year, and I had all kinds of brain tumor patients and stuff. I didn't have time to do anything else, but he had time to spend a lot of time in the lab the first year. Second year, then Lenny became my mentor. Now Lenny was an interesting guy too. He was also a New Yorker. I think these people were following me around. He was also from Brooklyn. He also had a little bit of a Brooklyn accent. And so he got along very well with Mike Martin. You know, they were like two peas in a pod. And turns out Mike's office was on the second floor of the institute, of the building, and the lab that assigned to him or that he had was in the basement. So I was 2 floors up. Well, Mike didn't spend a lot of time in the lab. He'd sort of come down every afternoon and shoot the breeze, asking us what we were doing and, you know, how was it going and so forth and so on. But he spent most of his time in his office, which was fine. Then I didn't know anything, you know. I was a complete greenhorn at that stage in my life. Some people say I still am, but so Len became my mentor, and he was my benchmate and my mentor. And he taught me everything in terms of the technologies that they were using in the laboratory. He taught me to draw blood. I knew how to draw blood, but we would draw a large quantity of blood, and we would separate then the red blood cells from the white blood cells, and we would count the white blood cells, and we'd put them into tissue culture media, and we could culture them, and they would survive for a while and so forth, and we could do various kinds of things to them. It turns out in 1960, you know, 10 or 11 years prior to that, a young fellow at the University of Pennsylvania by the name of Peter Knoll had discovered by accident, essentially, that if you do what we were doing, take the white blood cells, put them into culture, and add a substance that was called phytohemagglutinin. I think sometimes they make up these names so that it makes it hard for you to say them. Huh? Anyway, phyto meant plant, hemagglutinin meant clumping red blood cells or clumping blood cells. And people use this substance to clump up the red blood cells, and they would drop to the bottom of the tube then over about an hour or so. It was good, you know. And then you had in the supernatant or the plasma were the white blood cells, and then you could harvest them and do things with them and so forth. And Peter Knoll discovered in 1960 that if you put those cells into the incubator with this phytohemagglutinin and this stuff called, abbreviated again, PHA, that the lymphocytes would undergo a metamorphosis, just like me, and they would change from small, round, tiny little lymphocytes without much cytoplasm and not in a very dense nucleus. They would suddenly blow up like twice the normal size. They'd gain a lot of cytoplasm. Their nucleus would start to form all kinds of organelles in the nucleus that you could see under the microscope. And then they would duplicate their DNA and undergo mitosis. Well, that was a revelation in cell biology and also in immunology, because soon after 1960, people started trying to do the same thing with not with phytohemagglutinin, but with specific antigens. And an antigen in immunology is anything that stimulates an immune response. So we could put phytohemagglutinin in our cultures, and the cells would proliferate, and we could also stimulate the cells with specific antigen. And so the project that Michael wanted me to do would be to see whether or not I could generate proliferative responses on the part of normal lymphocytes and from normal people in response to antigens that we could show that the person had been exposed to before. For example, I started with mumps, and I could use the mumps vaccine, put it into culture, and the cells, some of the cells would recognize mumps and they would proliferate. It would take them a week to really reach the peak of their proliferative response, whereas with phytohemagglutinin, that happened in 3 or 4 days. And I couldn't understand why it took so long. I mean, the answer was essentially that the frequency of cells actually beginning to proliferate to the specific antigen was like less than 1%, whereas with the phytohemagglutinin, it was all of the T cells. All of the T cells would become activated and do their deal. So the question that we investigated was whether or not we could find out. So we had the history of the patient, if they'd had mumps vaccine or if they'd had mumps the disease. And we could also measure mumps antibodies in their blood. And we did the same experiments with rubella virus. Rubella virus caused German measles. And the reason that these two things, we could also look for their antibodies in their blood, and we also had the history of whether they'd been vaccinated or whether they'd have been infected with rubella and had the disease or not. The thing about both mumps and rubella, both the vaccine as well as the infection, last a long time, lifetime essentially. You get immunized as a kid, you might need a booster or something of that sort, but you get immunized as a kid and it lasts for the rest of your life. And of course, now in the days of coronavirus, everybody wants to know whether or not, if you get the infection, whether or not you're immune to a reinfection, which is a good question. And they're also wondering whether or not a vaccine, once we generate a vaccine, whether or not how long it will last, the immunity will last. And so that's the question that I started with in the laboratory in 1971, except with mumps and rubella. And I published 2 papers about that, some of my very first papers that said that the lymphocyte proliferation assay in response to these viruses was more sensitive than was the antibody test. And of course, that's what they're saying now with coronavirus. They're saying that, everybody, there's these Tcells, these cells that mature in the thymus. And I've talked about them in some of my other videos, but they name them Tcells because they come from the thymus gland, which is a big collection of lymphocytes in the upper chest. And so here we are 50 years later, and they're asking the same questions that I started with back in 1971. And it's a very interesting saga, essentially, that we'll get into as this book unfolds, because the purpose of this book, I think I said in some of the preliminary flyers about it, is that it's about, I'd like to educate people and expose them to what science is all about on the one hand, but also essentially focuses on immunology. Because for the most part, we don't get immunology taught in school unless you go to graduate school. You have to go all the way through high school and college and then go to graduate school in immunology. And then you get taught what I'm going to talk about in this series of videos. So we found that the T-cell lymphocyte proliferation assay was more sensitive in a better way, at least we thought, to detect prior exposure and reactivity and immunity to these viruses. And of course, you have to do that kind of an experiment with all of the different antigens that you might be interested in, tuberculosis, whooping cough, whatever, because each antigen is different in terms of the proportion of T cells that it activates in the very beginning. And once those cells are activated in the body, they do the same thing that they do in the tissue culture. They proliferate. So they expand markedly. I mean, logarithmic growth of cells that only recognize this particular antigen. Now that's important because that's the basis of immunity. That's the basis for why vaccination works. Because you vaccinate somebody with a vaccine that mimics the real microbe, and you get this expanded army of soldiers that are out there with fixed bayonets that are waiting around for the second time that you might be exposed to that antigen. And if you are, they basically trump on it right away. Those poor little microbes don't have a chance once the army is expanded. Now, and that leads to the next thing I wanted to mention in terms of this whole phenomenon is that, one of the things about coronavirus, I have to bring up the present point in time. One of the things about coronavirus is that a sizable fraction of the populace, mostly younger people, seem either they don't get any symptoms at all, even though they've been infected, or they have very mild symptoms. And as I've said before in my coronavirus videos, the symptoms are not due to the virus, presence of the virus. The symptoms are due to the immune response to the virus: fever, chills, malaise. All those non-specific symptoms are symptoms of inflammation going on in the body. So people have been wondering now, why are all these people sort of asymptomatic or only mildly symptomatic? And there's been reports out now within the last few weeks that indicate that, and lo and behold, there's other members of this mafia, this coronavirus mafia, that cause the common cold. 3 or 4 different viruses, and they call it they don't kill you like coronavirus can do, but they cause an upper respiratory infection that stays around for the symptoms stay around for a week or so, and then subside, and then you're fine. You can go back to work, you can go to school, and so forth. Well, the infection of the coronavirus, they've sequenced the genomes of these viruses, COVID-2, which is the one we're dealing with now, as well as the ones that cause the cold virus. And they've got a lot of, their sequences are very similar, which means that their gene products from those gene sequences will be very similar, which means if the immune system recognizes one of them, chances are the immune system is going to cross-react and recognize, virus number 2 that is not identical, but it's similar enough so that there is enough immunity and reactivity to deal with those with the newcomer. And I think that's what's really explaining things. And I think that the reason it's mostly the young people that are spared from really severe infection is because the young people have had more recent infections with the common cold viruses than maybe the old geezers like me that have been sequestered off in the nursing home for the last several years, and we haven't been exposed, not as recently as the younger people. So that's just one story that shows how the immune system works. The other thing that happened at the BCRC was that with Lenny Chess, he wasn't studying mumps or rubella. He was studying some compounds that were very, very interesting. And what they were synthetic molecules that had been made to try to mimic the genes of viruses. And in particular, they made double-stranded RNA molecules. There were 2 different varieties. One was called poly-AU, adenine and uracil, and the other one was poly-IC, inosine and cytidine. And so there was a long chain of inosine and then a long chain of cytidine, and they would wrap around one another and make a helix. And that was very similar to the morphology and of viral genes. And they had found, just prior to the time that Lenny and I were there at the BCRC, that these agents, if injected into experimental animals, those things could really boost up an immune response if you gave another antigen at the same time. And that's what we call adjuvants. So these things were really good adjuvants. And so Lenny took off, decided to see whether or not these agents would boost in vitro in our test tubes the proliferation of our lymphocytes, say in response to mumps or measles or whatever. And the answer was yes. So not only would they boost up immunity in vivo, they would do the same thing in vitro. And then Lenny and I collaborated, and I separated out the lymphocytes from the macrophages and treated each one of them with the poly-AU or the poly-IC and then let it incubate for a while. And then I washed those things off and we put the cells back together again. And, strange enough, it was the macrophages that were responding to the poly-AU or the poly-IC, and they were helping the lymphocytes to grow better. So we didn't know how that worked. We didn't have a clue about how that was going on. But that was a very interesting phenomenon because of the fact that we could sort of duplicate what was going on in the body. We could do it in the test tube, which allowed then for much more manipulation to be done than we could if we were just dealing with experimental animals and doing all the experiments in the animal. Well, all of these experiments basically showed that proliferation of these cells was fundamental for the generation of immunity. And they really, which was said already, which was already established essentially as dogma by Sir MacFarlane Burnet in 1957, who won the Nobel Prize in 1960 for enunciating the clonal selection hypothesis. And so what he proposed, and was subsequently found to be true, was that if you put an antigen into the body, they will seek out and find lymphocytes, T cells that have receptors for that antigen. And those are the ones that are selected to proliferate and expand. And so after all was said and done, as I studied more during that second year at the Baltimore Cancer Research Center, that this was a wonderful model system for somebody like me who was interested in cancer, trying to understand cancer, because I felt that was an awfully important problem. 50% of the people in the United States were dying from either heart disease or cancer, and nobody seemed to they didn't understand why that was. So, but I realized that you can't get at the cancer cell de novo. You've got to understand why the normal cell proliferates the way it does. And so this was a model system to be able to do that. So about towards the end of that second year at the BCRC, I had to make a decision as to whether or not I was going to stay there for a third year or move on. And this time then I had made my decision. I was not going to do medicine. I was going to do science. I really liked the whole scientific deal. It was a much more intellectual endeavor. And I just loved going to the library and looking up this paper and that paper and finding out what everybody was doing and so forth. Now, I could have stayed at the BCRC, but Lenny was moving on and he was my mentor, even though really Mike Martinet was my mentor, but he was upstairs and we didn't see him very often. And I felt that I needed, this is all in my really first year in the laboratory, I felt that I needed more hands-on mentorship. I needed somebody to take my hand and lead me through the process and so forth. And it seemed like that's what all young people that were wanting to become scientists, whether they were PhD graduates or medical graduates, they would spend several years in different laboratories, usually 2 years at a stretch, and doing postdoctoral work. And during those years is when they really learned and refined and defined their technological expertise and their craft and the whole deal. And so I wrote letters all over the country, trying to find another good place to go. Meanwhile, my wife and I and our kids, we lived in a little village north of Baltimore called Timonium, Maryland, in a brand new building, sort of a high-rise apartment building, which was fine. But Baltimore was a rough place back in the early 70s, probably way before that. The city was very segregated. It was divided up in pies, all over the circumference of the city, with an ethnic group in each one. The Poles lived in one section, the Jews lived in another section, and blacks in another. So it was quite an experience. And after having been at Yale, Yale New Haven Hospital, which was in New Haven, Connecticut, which was also a pretty rough city back in those days. And now with Baltimore, we felt like we wanted to get out into God's country. And so one of the places that responded positively to my letters seeking another postdoc was from Dartmouth Medical School, which is situated way up in New Hampshire, New England. And it's on the west side of New Hampshire, right on the border with Vermont, halfway north-south. And the Connecticut River is one bifurcates of those two states, New Hampshire on the one side and Vermont on the other. And towards the end of that second year at the BCRC, my wife and I we drove up there to interview to see what this place was all about. And that was in June. And of course, Baltimore, we've been in summertime since maybe the 1st of April because it was warm down there. And we got up to New Hampshire, and my wife had a sundress on. We walked out of the hotel, and we realized immediately we weren't dressed properly for this place, this northern climate. It was cold up there, like 40 degrees in the morning in June. And so, anyway, it was beautiful. Dartmouth is in Hanover, New Hampshire. That was named after the Hanover dynasty from the UK, as you probably know about those guys, George III and so forth. And there's Hanovers all over the country in different states and so forth. Anyway, we were there in Hanover, New Hampshire, and it was in the foothills of the White Mountains, rolling hills, very picturesque on the banks of the Connecticut River. Dartmouth was up on a big plateau overlooking the river. And it had a big town green with 17th and 18th century buildings facing the green. And so it was delightful. We fell in love, essentially. We decided we were going to go there no matter what. So we had to like this, the prospective new boss, who's a fellow by the name of Ross McIntyre, who originally was from Nebraska. And he sort of still had a little bit of the corn still in his hair, this guy, which I learned as time went on, One of his comments to me was that human beings can get used to anything. It just takes a matter of time. And no matter how bad it is, they can get used to it. His way of explaining to me that things could be, so they're not so good here at Dartmouth, but, whatever. Because the first day I walked in and presented myself as the new recruit at Dartmouth, I sat down with Ross McIntyre, and he said to me, well, there's good news and there's bad news. And I said, well, so give me the bad news first. So he said, well, I lost my grant, my research grant, and we have no money for this next year's experiments. And so I said to myself, oh, this is good. I didn't even know money was an important thing you had to have for science. So I left a place where money was no object. This was the United States government. Now we're spending trillions of dollars with just a drop of the hat. So I left a place with trillions of dollars, and I've gone to a place that had no money. So that was the bad news. The good news was that he could pay my salary because he got money from another source to pay my salary. But I didn't have a whole lot to do. On the other hand, there was Ross did have a clinical research project that was ongoing in his lab, and it fit in with what I had learned down in Baltimore with regard to the poly-AU and the poly-IC. Because at that time, in the early 70s, interferon was the hope for cancer. There was a lot of excitement about interferon. Interferon, I went through in one of the other videos, but interferon is an activity that is produced by cells when they're infected with viruses. And this activity sort of diffuses out of the cell and binds to receptors on neighboring cells. And as a consequence of that binding to the receptor on neighboring cells, those cells become immune to infection by the virus that's in the original cell. And that's why, so that this stuff interferes with the infection. So interferon was simply something that you could measure using a biological assay. It wasn't a real substance. There wasn't a molecule. You couldn't go to buy it from Sigma or get it from the NIH or whatever. Just, fill out an order form, send me some interferon. It didn't exist. It was just an activity. So what people did was, and Ross was one of them, he figured, well, maybe we can use interferon inducers. We can give that to cancer patients, induce interferon, they'll make it themselves, and maybe that'll have an anti-cancer effect. So he had already been in the process of doing these kinds of things a year before I got there. He had set up an interferon assay in the laboratory. And I learned how that was done. They wouldn't trust me with it, They wouldn't let me do the interferon assay. That had to be done by a technician who just did this thing full time, full stop. And I wouldn't trust this young clinical associate graduate to do these kinds of things. But I learned how it was done, and I watched them do it, and I analyzed the data. And it turns out that was very, very important for down the line, because the interferon assay was devised by virologists, and virologists were used to quantifying the amount of virus they had, say, in one test tube and comparing it to the amount in another test tube by doing titration curves. Now, a titration curve is when you dilute out. You take the tube or whatever it is, and you dilute it out serially over and over and over again. And then you do your assay. In this instance, you put in cells that could be infected by a virus. And you expose them to interferon, and then you came in and you infected all of the cells with the same amount of virus. And what you would find then was the cells that got a lot of interferon were protected from the infection. They didn't die, which is what you would measure. And the furthest that you could titrate out that sample meant, obviously, you had more interferon in that sample than maybe another sample where you could only do tenfold less titration. Now, that was very, very important, because that influenced me a great deal and it stood me in good stead for later on down the line. So what I did I was only there for a year at Dartmouth. And I forgot to say that one of the first things I did after McIntyre told me that he didn't have any money, I sat down and I wrote letters all over the world looking for a second postdoc, because the deal was, and with these kinds, I think, as I said before, usually the postdoctoral period was 2 years running. And so you would set these stagiaires up 2 years in advance. But I was so anxious to get started in learning how to become a scientist. I didn't want to wait around for 2 years and then go to another place. So I wrote letters all over the world. Particularly, I wrote to places in Europe, because I'd always wanted to study in Europe. And in particular, I wanted to go to France, because France is where all the good stuff is. They got the wine, they have the food, they have music, they have, it's everything. So I wanted to go to France. And it turns out that I was accepted almost by return post by a fellow in Paris which was good, by the name of Georges Maté, and he's right behind me here. I've done his portrait with his white coat on and so forth. And he was about 50 when I was there, and I was around 30. And so he accepted me to come to Paris. And so one of the first things I did back in Hanover in the fall, I enrolled in French 101 at Dartmouth College. So I was in this classroom with all these kids, these 18 years old. And I tried to not tell them how old I was. And when they finally found out, because we had to tell them how old we were in French, they gasped. The room gasped. They couldn't believe that this guy was in their class. But which would have been fine had I been a star student, but I was. I found the French grammar and the French pronunciation really difficult. I studied my tail off, but and I got was a little bit better by the time we finally went to France the very next year. And I did other things in Hanover and Dartmouth, one of which was to learn how to ski. And that was really a lot of fun. And so, and we had bought a house in Hanover because we couldn't find any, there weren't any apartments really to speak of. And so we had to borrow money from my wife's father, and we bought our first house, which was on almost an acre of land. And my wife was ecstatic because she could open the back door and let the kids run around. And so it was good. Everything was good except the job. You know, what are you going to do? And during this whole process, I'm becoming educated into the vagaries of so forth. You know, one of the things that happens to you as a person, you start in kindergarten and you go to school daily, every day, or every year, you change classes. So everything's prescribed for you. You know, you don't have to make any decisions, really. You just do what you're told. But if there comes a point in time whereby you have to take some responsibility for your life, and this was me finding out essentially the hard way that Michael Martinet told me not to go to Dartmouth. He said, "What are you going there for?" Just like that. He said, "That guy hasn't done anything." McIntyre meeting. McIntyre's claim to fame was that he was the second person after Peter Knoll to find that PHA would cause lymphocytes to proliferate. No, that was good, but that was in 1962. Now we're 10 years later in 1971 too. And McIntyre was spending most of his time in the clinic, on the wards, taking care of sick people, hematology patients and so forth. So that's just, in a nutshell, what happened to me. And, I forgot to tell you. I started to tell you, but I forgot to tell you that, so the clinical experiment that McIntyre was doing, back to that, he was giving poly-IC to cancer patients, and then he was taking blood from them at intervals thereafter and measuring interferon in the plasma. And then what I did, and that was done by that technician that wouldn't let me touch it. And then what I did was that I did lymphocyte proliferation assays to find out, you know, whether or not there was any influence by the interferon on the ability of the lymphocytes to proliferate. And we found that there was. And the interferon decreased the ability of the lymphocytes to proliferate, which was interesting. And it had been shown just almost simultaneously by a fellow in France in Paris by the name of Ian Gresser, who had shown that first, he had shown that interferon would inhibit the proliferation of cancer cells. And so then we found that it would inhibit the proliferation of normal cells. And that was interesting to me because I thought, well, maybe that's a handle on what's going on with both the cancer cells as well as the normal cells. So I learned a lot, even though it wasn't the most ideal situation. And so when the time came at the end of 1972, we packed up our belongings, and Lynn, my wife, had found that if we took the SS France to get across the ocean, we could take unlimited baggage. And of course, when we had 2 toddlers, I mean, can you imagine the stuff that we had to take with us. And so we had a stateroom at the bottom of the boat with no windows, no portholes, no nothing. If the ship had gone down, we would have never gotten off that boat. But it was fabulous. The food, we were able to, the kids ate before the adults. And so Lynn would go with the kids to have them eat beforehand. And they served the kids the same thing they served the adults. And Lynn would find out what was on the menu and what was good and so forth and so on while the kids were eating. And then the adults would eat. And so we had a great time. Lots of wine and whatever. It was really, really good. Normandy butter. Never forget. We landed so we took off. When we left New York on the 25th of June. Actually, that's in 72, it was 73. 25th of June. Now, that was the day that John Dean testified before the Senate and told them that there was a cancer in the presidency. All during this time, from like January on, we'd been undergoing the Watergate hearings. And we were becoming more and more and more disenchanted with the good old United States of America. And so we left dragging the flag and cheering our way across and drinking French wine across the Atlantic. That was good. And I'm going to stop there. That's the end of chapter 1.